Douglas from the Autonomic Center at Stanford University, and I'll be speaking on overlap between postural tachycardia syndrome, or POTS, ME/CFS, chronic fatigue syndrome, and long COVID and PASC, and what we can potentially learn from this overlap. These are my disclosures, none are relevant. So why are we talking about autonomic dysfunction in long COVID or PASC and chronic fatigue syndrome and other post-infectious illnesses in general? Well, many of the symptoms that we are seeing now in long COVID are those that we've seen for many years in patients with POTS. And some of these were discussed in the prior lecture, including many systemic symptoms such as fatigue, brain fog, GI issues, also pain syndromes that overlap with fibromyalgia, sleep issues, et cetera. And so this really heightened our interest in trying to understand this overlap, and with really the goal of understanding what causes autonomic dysfunction and debilitating disorders like POTS after infection. And before these associations were made, there have been many studies noting associations between chronic fatigue syndrome and autonomic dysfunction. Orthostatic intolerance is the most common symptom reported by patients, affecting at least 50%. There have been some studies looking at objective autonomic reflex testing in patients with ME/CFS using tilt table or heart rate variability analysis. Most of these studies have shown abnormalities, and the most common primary autonomic disorder that is diagnosed in these patients is POTS. It's present in up to 50% of patients, probably closer to 20 to 30%. And on the right here, this is a study just looking at autonomic symptom burden using a standardized symptom score called the COMPASS, showing that ME/CFS patients score very high on autonomic symptom burden. And these autonomic symptoms, the severity of these symptoms correlates with the severity of fatigue. There have also been several studies looking at cerebral blood flow in patients with ME/CFS, as well as long COVID, which we'll touch on. And these studies come out of Campen group using measures of cerebral blood flow. And you can see here on the graph on the left here, compared to controls, which on tilt testing demonstrated just a 7% reduction in cerebral blood flow, which is normal. Patients with ME/CFS on average had around between 24 and 29% reduction in their cerebral blood flow. And this was regardless of whether or not they had POTS or not. So 28% of patients in this study with ME/CFS met criteria for POTS, and 58% had normal blood pressure and heart rate on tilt testing. However, this was sort of independent of the reduction in cerebral blood flow. So even in patients with only orthostatic intolerance symptoms not meeting POTS criteria, there was still a drop in cerebral blood flow. There's also been studies looking at carbon dioxide measurements and tidal CO2, showing that patients with ME/CFS have, in some cases, a baseline hypocapnia as well as a hypocapnia with upright positioning. As we know, hyperventilation can reduce CO2, which can also affect cerebral blood flow and causing vasoconstriction in the intracranial arteries. However, there have been studies that have suggested that the reduction in blood flow is independent of the hypocapnia or hyperventilation in these patients. So the same group did a follow-up study just looking at long COVID and comparing that to ME/CFS, showing similar reductions in cerebral blood flow. So the phenotype seems to be similar, at least in what was seen with the blood flow measurements. There's very, very similar reduction in cerebral blood flow on the orthostatic challenge in those with long COVID compared to those with ME/CFS. So switching gears just to long COVID and this association with POTS. Well, very early on in the pandemic, we saw this. There's now been many case series published. The themes that emerge are that most patients are female. Most had pretty mild initial COVID-19 symptoms. And we see very, very prominent cognitive fatigue, orthostatic intolerance, as was described earlier. If we ask closely in seeing these patients, we get a history that probably was some mild pre-existing self-limited autonomic symptoms, and the virus perhaps just sort of augmented those and really, brought out a much more severe phenotype. So we tried to get at how severe or common autonomic dysfunction is in long COVID using a standardized autonomic score, symptom score, the COMPASS. And we found that almost about 2/3 of patients scored in the moderate to severe range on this autonomic scale. And most of our patients were non-hospitalized. And we really saw no difference in total autonomic burden comparing hospitalized and non-hospitalized, suggesting that the initial infection severity is independent, of the likelihood of developing long COVID or autonomic impairment from this. If we put these numbers in perspective, 67% of patients that we surveyed, this would translate into somewhere between 5.9 to 17.8 million patients in the U.S. population, depending on how we calculate prevalence of long COVID, that might have some significant autonomic burden after a COVID infection. So looking at what pre-existing conditions might predispose one to develop, more severe autonomic dysfunction, we saw common themes that have been reported in other studies, pre-morbid asthma, obesity, any autoimmune disease, anxiety, depression, and history of smoking, all predispose patients to more severe autonomic dysfunction. So those are just a snapshot of some symptom cross-sectional studies. Now, what about sort of deep phenotyping? Well, this was really, I think, the first study that came out looking at objective autonomic testing in patients with long COVID. And this is out of the Brigham group, where they took 9 patients with PASC, and this is not necessarily POTS, just long COVID, comparing them to patients with POTS that did not have COVID and pre-existing database of healthy controls that they had. And what they did was they did the standard autonomic testing that we do, and they also measured cerebral blood flow and CO2 until. And what they found was 100% of PASC patients had abnormalities. These sort of spanned all the domains that we can test on autonomic reflex testing, including sweat dysfunction, parasympathetic dysfunction, and really prominent sympathetic adrenergic dysfunction. So, and then when they looked at cerebral blood flow, which we could see here on the right, in controls, which is in green here, the controls had a normal reduction in cerebral blood flow on tilt. Patients with POTS not due to COVID had a very significant reduction in cerebral blood flow, and patients with PASC had an even more significant reduction in blood flow on tilt, sort of confirming what the prior studies from the Campen group have confirmed in MECFS and also in long COVID. So now when they also looked at CO2, they found that patients with long COVID had a more severe reduction in cerebral blood flow when correcting for CO2. Because as we know that with hypocapnia, which you can see with hyperventilation, which many of these patients have on tilt, that will further reduce your cerebral blood flow potentially. So correcting for that, they found that PASC patients or long COVID patients had 100% of them had abnormalities, i.e. hypocapnia on tilt, compared to 75% of POTS. And the correcting for this hypocapnia in the long COVID group showed that patients had a more severe reduction in cerebral blood flow. So sort of take home, we see reduction in blood flow to the brain in both long COVID and POTS independent of long COVID, as well as MECFS independent of long COVID. And we don't always see a tachycardia on tilt or a abnormal blood pressure reduction on tilt. So just looking at heart rate and blood pressure as we do kind of in our standard autonomic lab is really not sufficient now. I think in the future, I think pretty much all labs should be thinking about measuring cerebral blood flow as well as end-tidal CO2. And this is just recently, I saw this company, biotech company is looking at wearable monitors of cerebral blood flow that just fit in the ear that can measure heart rate, blood pressure, and cerebral blood flow that one would wear continuously, really targeting the long COVID population. So I think being able to measure this with wearables over long periods of time is going to be, very interesting. We're going to see this in the near future. So we're also doing our own study looking at similar metrics. We're also measuring cerebral blood flow, as we can see, as well as near-infrared spectroscopy, which is a surrogate of oxygen extraction, as well as norepinephrine levels on supine and upright tilt, CO2 measurements, and we're also doing skin biopsy to look for small fiber nerve density, which is a surrogate for small fiber neuropathy, as well as something called alpha-synuclein, which I'll show you in a minute. So preliminary data, we've tested 18 patients so far compared to 8 controls, similar age and gender. Not surprisingly, patients with post-COVID POTS, and I should mention in our study we're really targeting POTS that's developed from long COVID or post-COVID POTS, and not long COVID in general, but really just trying to get at what causes POTS after long COVID. So not surprisingly, the post-COVID POTS patients had tachycardia on tilt, whereas controls did not. They also had higher blood pressure on tilt, which is the theme that we're seeing, that these patients have what we call a hyperadrenergic state, which may or may not be due to elevated norepinephrine. And then 75% of our patients had abnormal reduction in cerebral blood flow, confirming prior studies, and most patients had objective autonomic dysfunction that spanned multiple domains. We have not found really abnormalities in end-tidal CO2, measures of oxygen extraction, or norepinephrine levels so far. Granted, these are small sample sizes. So on skin biopsy, so the Brigham group also performed skin biopsy, and they found very high percentages of small fiber neuropathy. We found about 43% of our patients have small fiber neuropathy, but a finding that we were not anticipating was 36% of our patients have phosphorylated alpha-synuclein in the nerve fibers, and phosphorylated alpha-synuclein is what we commonly see in Parkinson's disease. And so, the relevance of this finding within long COVID is not clear. Are these patients just patients that would have developed Parkinson's or will develop Parkinson's disease down the line? Is this an incidental finding, or does SARS-CoV-2 somehow induce deposition of this Parkinsonian protein and therefore increased risk of Parkinsonian syndromes? Follow-up will be necessary to really answer this question. So in the last few minutes of my talk, I'm going to try to just give a potential overview of mechanisms that could underlie these post-infectious syndromes, really focusing on long COVID, but I think this extends to both POTS and ME/CFS unrelated to long COVID. And many of these will be discussed in further detail in this lecture. So one thing that we're really looking at is immune dysregulation. We see that our patients with POTS, many of them have underlying autoimmune disease, or some of them have elevated inflammatory markers. There is a potential mechanism involving vagal nerve damage. We've seen several recent studies demonstrating SARS-CoV-2 in the vagus nerve. And there's also this body of literature in the POTS literature that has shown elevated G protein coupled receptor autoantibodies in patients with POTS. These autoantibodies may target autonomic receptors. However, whether they're causative or this just a bystander effect is unclear. And so these studies are ones that have shown sort of elevated autoantibodies in patients with long COVID. So it's not just GPCR autoantibodies, but really a host of potential autoantibodies. And there are many groups now really doing extensive work in this. And I think the RECOVER NIH study will be helpful in sort of really elucidating some of these antibodies. Of course, viral persistence. We know many studies now that the virus can persist for many, many months, whether in gut reservoirs or elsewhere in the body. Just as viral persistence is a potential mechanism in ME/CFS. And whether or not this virus can persist in autonomic ganglia is an interesting question that has not been answered. And viral persistence is really, I think, the target of this initial wave of research and out of the RECOVER trial and clinical trials. Mast cell activation, many of our patients with POTS, with ME/CFS, have this elevated, mast cell hyperactivity where they'll get flushing and, many new onset allergies. And interestingly, some of these GPCR autoantibodies can also act on mast cells to stimulate early maturation. So, the interplay of the autoimmune disease and hyperactive allergic syndromes and autonomic dysfunction is very interesting and I think a very tight association. There's also a lot of our patients have big swings in blood pressure and heart rate, not just POTS, but also orthostatic hypertension. And baroreflex impairment could also be a potential autonomic mechanism. The virus could potentially invade some of these nerves and also areas of the brainstem that modulate blood pressure and heart rate. Deconditioning, many of our patients are deconditioned. However, deconditioning doesn't explain the whole host of symptoms that we see in these disorders. Exercise can be a very important treatment, but we have to be very, very wary of post-exertional crashes, which we always discuss in detail with our patients. And so in considering treatment, first we really want to stabilize the symptoms and then introduce exercise very gently in a very individualized fashion. And then finally, gender physiology. So, many of our patients, with POTS and long COVID, I think are women, are female.